Hello students, welcome back to a new video of Sunanda's tutorial. In this video we will discuss the experiments to prove DNA as the genetic material. In my previous video I have discussed about Frankel-Conrad experiment. In that experiment RNA was proved as the genetic material of RNA viruses. Today we will discuss different experiments like Griffith experiment, Horsey's experiment to prove DNA as the genetic material. So let's start. At first, DNA or deoxyribonucleic acid, which is the carrier of all genetic information, it codes genetic information passed on from one generation to another and determines individual attribute like eye color, facial features. All our characters are controlled by this DNA or genes. Genes are actually DNA. Although DNA was first isolated in 1869 by Swiss scientist Friedrich Miescher from nuclei of pus-rich white blood cell, he called it nuclein. At that time, DNA was not named and termed DNA. So he called this as nuclein. And its role in the inheritance of traits was not realized until 1943. But it was first isolated by Swiss scientist Friedrich Miescher and he called this DNA as nuclein in 1869. Then one experiment was conducted by Griffith in 1928 before Friedrich Miescher. Friedrich Griffith was first carried out the first experiment suggesting that bacteria are capable of transferring genetic information through a process called transformation. So it is a type of horizontal transfer. Okay. And he told that he took two strains of pneumococcus or streptococcus in his experiment. So he used two strains of pneumococcus or streptococcus, Streptococcus pneumoniae S3 strain or smooth strain and R2 strain. So he took two strains R2 and S3. S3 strain is called smooth strain having capsule on its cell and R2 is called a rough strain as it have no capsule on their cell. Smooth strains are virulent. But these R2 strains are not virulent. This virulent strain can cause pneumonia disease, but this R2 strain cannot cause pneumonia. Okay. Then he did his experiment. He took 4 situations. At first, he injected S3 type of strain into the mice and the mice died due to pneumonia. Okay. Then he injected R2 type of bacteria into the mice, but the mice lived and pneumonia was not produced. Because R2 strain are not virulent, these are avirulent. Then he injected heat-killed S3 type of strain into the mice. The mice lived and pneumonia was not produced. In case of heat-killed, they just removed or destroyed the capsule layer of that strain. So mice lived. Then he mixed R2 type of bacteria and heat-killed S3 type of bacteria and togetherly injected into the mice. Then he observed that the mice died due to pneumonia and virulent S3 type living bacteria could also be recovered from their body. Okay. In the last case, he took living R2 strain and heat-killed S3 strain togetherly and injected into the mice, but he observed that the mice died due to pneumonia and virulent S3 type of bacteria also recovered from their bodies. So this fourth part of the experiment indicates that some R2 type of bacteria or avirulent bacteria were transformed into S3 type of bacteria. This occurred perhaps due to the absorption of some transforming principle by R2 type of bacteria from heat-killed S3 type of bacteria. And this transforming substance from S3 type of bacteria caused the synthesis of capsule, which resulted in production of pneumonia and death of the mice. And Griffith called this as transforming. So Griffith termed this as transforming principle, and the transforming principle to that substance which was transferred from S3 to R2, making it living S3 type of bacteria. Okay. Based on the above result, he inferred that something must be transferred from S3 strain into non-virulent R2 strain bacteria that transformed them into smooth coated. So something is transferred from S3 to R2 type of bacteria so that they transformed into S3 type of bacteria. So the material that was transferred referred to as transforming principle. Okay. Next. After Griffith, Avery, MacLeod and McCarthy experiment. So later on in 1944, they did the experiment, repeated this experiment in lab in vitro to identify that transforming substance, and they proved that this transforming substance in fact the DNA. Okay, so they prepared culture containing heat killed estrogen and removed the lipid and carbohydrate from the solution. Next, they treated the solution with different digestive enzymes like DNase, protease and RNase. DNase can destroy DNA, RNase can destroy RNA, protease can destroy proteins. Finally, they introduced living R strains cells to culture, which culture would develop transformed S strain bacteria. Only in the culture treated with DNase did the S strain bacteria fail to grow. This indicated that DNA was the genetic component that was being transferred between the cells. So at first they destroyed all the proteins, RNAs, carbohydrate, lipids, but none of the situation can affect the process. But when they used DNase to destroy the DNA, then the whole process was affected. So they concluded that this transforming principle is actually the DNA, not protein, not RNA, not lipid, not carbohydrate. It is the RNA which caused this transformation in bacteria. Then Horsey-Chase experiment. So it is another evidence that DNA is the genetic material, and it was proved by Alfred Horsey and Martha Chase in 1952. Okay. Their experimental tool was bacteriophage virus that attack bacteria which specifically involved in infection of Escherichia coli with bacteriophage. So what they took bacteria E. coli and bacteriophage virus, and they conducted their experiment in 1952. Okay. So at first bacteriophage was allowed to grow. Bacteriophage virus was allowed to grow on two of the medium. one containing radioisotope phosphorus or P32. Radioactivity means simply we have to tag something so that we can trace. Okay, and another containing radioactive isotope of sulfur. One medium containing radioactive isotope of phosphorus P32, another medium containing radioactive isotope of sulfur S35. Phages grown on radioactive phosphorus contain radioactive P-labeled DNA, as DNA contains phosphorus, not sulfur. So phages that grown on that radioactive phosphorus containing medium, they contain P-labeled DNA, as DNA contains phosphorus. Phosphorus or phosphate is a part of DNA but not sulfur. Similarly, viruses grown in the medium containing radioactive sulfur contain radioactive S35 labeled protein, because sulfur is found in many proteins but absent in DNA. Phosphate is the part of DNA, but sulfur is not the part of DNA, it is the part of protein. Then E. coli is the bacteria were introduced to be infected by the radioactive phase. After progression of the infection, blender was used to remove the remains of phages and phase part from outside the bacteria, followed by centrifugation in order to separate bacteria from phase debris. Actually, when E. coli or bacteria is infected by this phage virus, they only insert their DNA inside the bacteria. All other parts regarded as phase debris, they remain outside of the E. coli cell. So during the process of centrifugation, the bacteria and the phase debris will be separated. So centrifugation results in settling down of heavier particle like bacteria in the form of pellet, while those light particle such as medium, phase debris, phase part float near the top of the tube called as supernatant. Okay, so they observed that on measuring the radioactivity of the pellet and supernatant in both media, P32 was found in large amount in the pellet, where S35 is present in supernatant, because in pellet bacterial cells are present in the pellet, and bacterial cells contain DNA, and that DNA have P32, and in supernatant part they have proteins and other phase parts, phase debris, where proteins are present because the outer capsule of that phase contains protein, and that protein contains S35. So the conclusion of this Hershey-Chase experiment was, they deduced that it was DNA, not protein, which got injected into the host, and thus DNA is the hereditary material that is passed from virus to bacteria. So these are 3 experiments which showed that DNA is the genetic material. I hope you really enjoyed this session, will get benefit from it. In my next class I will discuss some other important topic of botany. So please stay tuned and enjoy my videos like this. Thank you.